Welcome to edupediaworld.com. This is Somcha J. Nair, your online biology tutor. In the last video, we discussed about the structure of DNA and RNA. We also discussed about discovery of structure of DNA by Watson and Crick and different experimental proof for that. Here, we are discussing about experiments which prove DNA as genetic material and about DNA replication. Even though the discovery of Maclyn by Vicia and the proposition for the principles of inheritance by Mendel were almost at the same time, but that the DNA act as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt, Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most of the cells. But the question of what molecule was actually the genetic material had not been answered. The transforming principle, which actually proved the DNA as the genetic material. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumonia, that is the bacterium which is responsible for causing pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism, that is the bacteria, had changed its physical form. When Streptococcus pneumonia, or otherwise called pneumococcus, bacteria are grown on a culture plate, some produces smooth, shiny colonies that will be denoted with capital letter S, while the other produce rough colonies which is denoted by capital letter R. This is because the extrained bacteria have a mucous polysaccharide coat, that is a polysaccharide coat, that mucous coat is a polysaccharide coat over the bacteria, while the R strain doesn't have that polysaccharide coat. Mice infected with the S strain, that is virulent, that S strains are virulent because it have the polysaccharide coat, die from pneumonia infection, but the mice infected with the R strain do not develop pneumonia. Griffith was able to kill bacteria by heating them. He observed that the heat killed the S strain bacteria injected into mice did not kill them. When he injected a mixture of heat killed S and live air bacteria, the mice died. Moreover, he recovered the living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle, that is transferred from the heat killed S strain, had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent in nature. This was due to the transfer of the genetic material. However, the biochemical nature of the genetic material was not defined from his experiment. Here you can see the pictorial representation of Griffith experiment. Here you can see that the rough strain is injecting to the mice, but the mouse is live. When the smooth strain is injected, the mouse dies. When the heat killed smooth strain is injected, the mouse is live. When the rough strain plus heat killed smooth strain is injected, the mouse dies again. Biochemical characterization of the transforming principle. This experiment actually proved that DNA is the genetic material. Prior to the work of Oswald, Avery, Colin, MacLeod and McCarty from 1933 to 1944, the genetic material was thought to be a protein. They worked to determine the biochemical nature of the transforming principle in the Griffith experiment. They purified biochemicals like proteins, DNA, RNA, etc. from the heat-killed S cells to see which one could transform the live R cells into the S cells. They discovered that DNA alone from the S bacteria caused R bacteria to become transformed. They also discovered that the protein digesting enzymes, proteases, and RNA digesting enzymes, RNases, did not affect the transformation, so that the transforming substance was not a protein or RNA. digestion with DNases did inhibit the transformation, suggesting that DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced in this case. Here we discussed about the DNases. Then what is DNases? DNases is the deoxyribonuclease. Is an enzyme that catalyzes the hydrolytic cleavage of the phosphodiester linkages in the DNA backbone, thus degrading the DNA. Deoxyribonucleases are one type of nuclease. It's a generic term for enzymes capable of hydrolyzing phosphodiester bonds that link to the nucleoids. This is a pictorial representation of the biochemical characterization transforming principle, that is the Avery, MacLeod and McCarty experiment. The Hershey-Chase experiment, the unequivocal proof that the DNA is the genetic material, came from the experiments of Alfred Hershey and Martha Chase in 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was a protein or DNA from the viruses that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contained radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in the blender. So this experiment also called as blender experiment. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with viruses that have radioactive DNA were radioactive in nature, indicating the DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with the viruses that had radioactive proteins were not radioactive. This indicates that protein did not enter the bacteria from the virus. DNA is therefore the genetic material that is passed from virus to bacteria. This is a pictorial representation of Hershey and Chase experiment. Here you can see that the viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, but the DNA contains phosphorus but protein does not. Similarly, the viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. These phages are allowed to attach to E. coli bacteria, then the infection is happening. Then the viral coats were removed from the bacteria by agitating them in the blender. Then the particles were separated from bacteria by spinning them in a centrifuge. Then bacteria which was infected with the viruses that had radioactive DNA were radioactive in the nature, and which is infected with the radioactive protein ones have no radioactive in the supernatant. And this indicates that DNA is the genetic material. From the foregoing discussion, it is clear that the debate between protein versus DNA as the genetic material was unequally resolved by Hershey-Chase experiment. It became an established fact that it is the DNA that is genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic viruses, QB bacteriophage, etc. Properties of genetic material. A molecule that can act as a genetic material must fulfill the following criteria. It should be able to generate its replica through replication. It should chemically and structurally be stable. It should provide the scope for slow changes like mutations that are required for evolution. It should be able to express itself in the form of the Mendelian characters. Why DNA is the predominant genetic material? The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in the physiology of the organism. Stability as one of the properties of the genetic material was very evident in the Griffith transforming principle itself, that the heat which killed the bacteria at least did not destroy some of the properties of the genetic material. This now can easily be explained in light of DNA, that the two strands being complementary, if separated by heating, coming together when appropriate conditions are provided. Further, 2'OH group present at every nucleotide in the RNA is a reactive group and makes RNA labile and easily degradable. RNA is also known to be catalytic in nature, hence very reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, DNA is considered as the better genetic material. In fact, the presence of thymine at the place of uracil also confers an additional stability to DNA. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate compared to DNA. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve very faster compared with the DNA as the genetic material. RNA can directly code for synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for the synthesis of protein. The protein synthesizing machinery has evolved around RNA. The above discussion indicates that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information, and for the transmission of genetic information, RNA is the better one. RNA world. From the foregoing discussion, an immediate question becomes evident. Which is the first genetic material in the nature? RNA was the first genetic material. There is no enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst and not by protein enzymes. But RNA being a catalyst was very reactive in nature and hence very unstable. Therefore, DNA has evolved from RNA with chemical modification that make it more stable in nature. DNA being double-stranded and having complementary strands further resists to changes by evolving a process of repair. Replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: "It has not escaped our notice that the specific pairing we have published immediately suggests a possible copy mechanism for the genetic material." By Watson and Crick in 1953. The scheme suggested that the two strands would separate and act as a template for synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This is termed as semi-conservative DNA replication. Here you can see the pictorial representation of a DNA replication. The experimental proof for replication. It is now proven that the DNA replicates semi-conservatively. It was shown first in E. coli or Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Meselson and Franklin Stahl performed the following experiment in 1958 to prove that. They grew E. coli in a medium containing ammonium chloride, that is 15NH4Cl. That N15 is a heavy isotope of nitrogen. As the only nitrogen source for many generations, the result was that the nitrogen was incorporated into the newly synthesized DNA as well as the other nitrogen containing compounds. The heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. Please note that nitrogen 15 is not a radioactive isotope. It can be separated by 14N, that is the nitrogen 14, only based on the densities. Then they transferred the cells into a medium with normal ammonium chloride and took samples at various definite intervals as the cell multiplied and extracted the DNA that remained as double-stranded helix. The various samples were separated independently on cesial gradient to measure the densities of DNA. Thus, the DNA was extracted from the culture one generation after the transfer from 15 nitrogen to the 14 nitrogen, that is the nitrogen N14 medium. That is after 20 minutes. E. coli divides in every 20 minutes. This had a hybrid or intermediate density. The DNA extracted from the culture after another generation, that is after 40 minutes, that is the second generation, was composed of equal amounts of this hybrid DNA and light DNA. This is the pictorial representation of Meselson Stahl experiment. Here you can see that the bacteria is first grown in the heavy medium with ammonium chloride which have N15, and then they transferred it into the light medium which have N14 in it. Then they have taken samples at 0 minute, samples after 20 minutes the first generation sample, the sample after 40 minutes the second generation sample. Here you can see that the sample at the 0 minute with have whole heavy chain, that is the DNA is heavy because it have the N15, and in the second it is the first generation that have intermediate band. It can one strand with heavy nitrogen and one strand with light one, that is the N14, and the second generation half is intermediate and half is light. Proof for replication in Vicia faba. Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vicia faba, the faba beans, by Taylor and colleagues in 1958. The experiments proved that DNA in chromosomes also replicate semi-conservatively. The enzymes involved in the replication. In living cells such as E. coli, the presence of replication requires a set of catalysts, that is called enzymes. The main enzyme is referred to as a DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli has only 4.6 into 10 raised to 6 base pair. Compare it with the human whose diploid content is 6.6 into 10 raised to 9 base pair. Complete the process of replication within 38 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with a high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleoside triphosphates serve a dual purpose. In addition to acting as a substrate, they provide energy for polymerization reaction. The two-terminal phosphates in the deoxynucleoside triphosphates are high energy phosphate, same as ATPs. The machinery which is involved in the replication. In addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of replication with very high degree of accuracy. For long DNA molecules, since the two strands of the DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix, referred to as the replication fork. The DNA-dependent DNA polymerase catalyzes the polymerization only in one direction, that is from 5' to 3'. This creates some additional complications at the replication fork. Consequently, one strand, that is the template strand with polarity 3' to 5', the replication is continuous, while on the other, the template with the polarity from 5' to 3', it is discontinuous in nature. The discontinuous synthesized fragments are later joined by the enzyme DNA ligase. The DNA polymerase on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. This is a definite region in E. coli DNA where the replication originates. Such region is termed as the origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedure, requires a vector. The vectors provide the origin of replication. In eukaryotes, the replication of DNA takes place as S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy or chromosomal anomaly. Here I am going to summarize the DNA replication process by the pictorial representation of it. You can see that the enzyme helicase is unwinding the parental double helix. This enzyme is responsible for breaking the hydrogen bond and separate the double-stranded DNA into single strand. After it becomes single strand, the single strand binding protein will come and stabilize the unwound parental DNA. Otherwise, it will go back and again hybridize to form DNA double helix. Then the leading strand is synthesizing continuously in the 5' to 3' direction by the DNA polymerase. I already told that DNA polymerase only synthesizes from 5' to 3' direction. So, in the lagging strand, it synthesizes it discontinuously, not continuously. Primer synthesizes a short RNA primers which is extended by DNA polymerase to form an Okazaki fragments. The short discontinuous DNA is called Okazaki fragment. After the RNA primer is replaced by DNA by another DNA polymer which is not shown here, it will join it and the DNA ligase join each Okazaki fragment to the growing strand. Thus, I am concluding this video. Thank you. In the next video, we are going to discuss about transcription.